Let me look. Maybe I don't. I'll have to find the better picture of it. Basically, what we're doing when we're using enzymes is we are lowering what's called the activation energy. So these chemical reactions are occurring in organisms, and they're either releasing energy or they're consuming energy. In either case, the reaction needs a little push, and that push is called activation energy. So these enzymes act as a catalyst. Catalyst means to speed something up. So they speed up these reactions through lowering the activation energy. So what we see in this first graph right here is that the rate at which an enzyme catalyzes a reaction, or basically the speed that the reaction happens, is influenced by certain chemical and physical factors. And so this has a whole bunch of words on it here, but I circled the ones that are important. So here first we have enzyme activity is being influenced by enzyme and substrate concentration. So the reaction rates increase with an increased amount of enzymes. And so we have a certain point that we hit, though, where pouring more enzymes onto the substrate, no more are needed, and so the additional enzymes no longer increase the reaction rate. So it speeds the reaction up into the point where everything has been used, and then adding more enzymes is no longer necessary. So the rate at which the enzyme catalyzes a reaction is influenced by 1, the increased amount of enzyme, 2, the temperature. And so reaction rates are increasing at higher temperatures, but only up until the optimum temperature. So if you think about the human body, our body is going to do things and go through its chemical reactions up until it hits 98.6, and then as you start creeping up higher and higher, you get to that point when you have a fever, and if you have too high of a fever, you're not going to be able to survive that, because fevers are going to denature the proteins that are inside of your body, and once you do that, you have denatured proteins, everything starts to break apart, and that's why people get sick and die after too high of a fever. So we have the amount, we have the temperature, we have the pH. So your body has an optimal pH where your blood is going to remain stable. You've got this optimum level for each one of the enzymes. So the optimal level in your blood is going to be different than the optimal level in your stomach. Every part of the body has this optimal pH where above or below it, if you're below it, it happens too slow. You get to this point, which is the optimal, the best pH, the peak pH. And then once you get past that point, it's very sharply going to dip off, and then eventually pH is not going to help anymore. So optimal temperature, optimal pH, and then finally the presence of what's called an inhibitor or an activator. So reaction rates increase in the presence of activators. So here we have our activator that's making our chemical reaction going slow, slow, slow. All of a sudden it speeds way up. It gets up to the optimal level. And then if we have an inhibitor, it's going to stop the reaction from occurring very, very quickly. And so enzymes, if you think about some of the processes that involve enzymes, things like the turning of leaves in the fall. Once we get to fall temperatures where it's no longer summer outside and the temperatures are cooling off, then leaves start to turn yellow and red and orange and eventually start to turn brown and die off because of enzymes. That's also what happens in your fruit ripening, in your food digesting. All kinds of different processes are controlled by enzyme activity. The best one that I can think of for activator, if you've ever used a meat tenderizer to help make your steak taste better when it comes off of the grill because it's nice and soft and and juicy. That would be an example of an activator. So to go back to the slide that I skipped, these enzymes are proteins that initiate and speed up chemical reactions. So enzymes are catalysts. A catalyst is something that speeds up these chemical reactions. Enzymes are not permanently altered in this process, but rather they can be used over and over again. So the enzymes inside of your body, they go in like a lock and key. They open up what they're supposed to open up. The chemical reaction occurs, and then they move on to the next reaction. So if I can move past that. Sometimes what we find, however, is that we have what's called a misspelled protein. So basically, if you think back to your DNA mutations, if you have a mutation, then something really bad happens. If we have a mutated or a misspelled protein, we have an incorrect amino acid sequence. So basically, these amino acids are being put together, and they're put together. This one's a functioning enzyme, and this one is a non-functioning enzyme. So this one spells out the correct code for the protein. If, however, we have an incorrect or non-functioning enzyme, then that's what causes a lot of different diseases and a lot of different physiological problems, or the ability of your body to function. So we have things like, here they talk about phenylketonuria. It's hard to say. Phenylketonuria, that's if you find Diet Coke really bitter, then phenylalanine is the enzyme that you taste. And so that's one that wouldn't cause any kind of debilitating disease. But people who can't taste it, they have the functioning enzyme. People who taste that really bitter taste, they have the non-functioning enzyme.